Okay, so the first question that we have for our present topic on transportation in plants and photosynthesis. So the first question that we have is amount by which water potential is reduced due to presence of solute is called as. What is this amount by which water potential is reduced due to presence of a solute? What is it defined as? Is it called as pressure potential, solute potential, metric potential, or none of this? So the answer is it is solute potential. So whenever the solutes are added, the water potential is going to get reduced. So we call this as solute potential. So this is the definition of solute potential. So the most abundant solute in phloem sap is, this is very easy. Sucrose is the most abundant solute in phloem sap. So the third question, so statement question it is, assertion is if a pressure greater than atmospheric pressure is applied to pure water or a solution, its water potential increases. So water potential of a cell is affected by both solute and pressure potential. So both the statements are correct, but reason is not the correct explanation of A. So B is the answer for this. Both A and R true, but R is not the correct explanation of A. So water potential of a cell is affected by both solute and pressure potential. It's a right statement, but that is not an reason for the assertion statement. Pressure potential of a plant cell and its Targar pressure are. So pressure potential of a plant cell and its Targar pressure are numerically equal, but first is in negative and second in positive value. Numerically equal and both are positive. See, another name for Targar pressure is pressure potential. So numerically it is equal and both are positive. So B is the answer for fourth one. So the pressure potential of a plant cell and its Targar pressure are numerically equal and both of them are positive. Okay. So fourth one, the answer is B. Fifth one, the loss of water in liquid phase from the leaves is called guttation. Guttation takes place at night only. So that is the fifth question. So you have to find out which is right. Assertion is true, but reason is wrong because even in the early in the morning, guttation takes place. So it is not just at night. So assertion is true, but reason is false. So C is the answer for fifth question. Then sixth one, which of the statement is not correct? That implies three statements are correct and one is wrong. The rate of absorption of water is almost directly proportional to the rate of transpiration. Soil temperature, soil aeration, relative humidity, amount of soil water, and transpiration are factors which affect the absorption. Water is removed in the form of vapors during transpiration. If the atmosphere is humid, it increases the rate of transpiration. This is the wrong statement. If the water atmosphere is humid, it decreases the rate of transpiration. So D is the wrong statement for the sixth question. So D is the wrong statement. So which contributes most to the transport of water? So from the soil to the leaves of the tree. So which of them contributes the most to the transport of water from the soil to the leaves of a tree? Root pressure, coefficient of water and transpirational pull, capillarity rise of water, or hydrolysis of ATP. B is the right answer. It contributes cohesion of water and transpirational pull contributes the most for the transport of water from the soil to the leaves of a tree. So forces generated by transpiration can create pressure sufficient to lift water even up to height of 130 feet, 130 meter, 230 feet, 230 meters. So the answer is 130 meter. Even in your textbooks, the same concept has been given that pressure is sufficient to lift water up to the height of 130 meter. Of all the environmental factors, which is the most influential in determining the rate of transpiration? So is it light, water, relative humidity of atmosphere, or temperature? So which is the most influential in determining the rate of transpiration. So there are four factors. So which one is the most influential? So the relative humidity of atmosphere is the most influential. So if humidity is more, even though light, water, temperature are all favorable, so there is going to be no transpiration will be lower. If humidity is less, transpiration will be higher. So the most influential in determining the rate of transpiration is relative humidity of atmosphere. Okay. So the 10th question, the process which regulates the turgor of cells of leaves and prevents it from becoming high, okay, is what is that cell? Process. So turgor of cells of leaves and prevents it from becoming high is transpiration, photosynthesis, respiration, guttation. So which process is going to regulate the turgor of cells of leaves and prevent it from becoming high? So which of that is the process? So 10th one, the answer is A, transpiration. So transpiration is going to prevent uh, the turgor of cells of leaves to become very high. Okay. Water absorbed by root hairs of a plant can rise to the highest point by means of root pressure, imbibition, transpirational pull, or capillarity. The answer is transpirational pull. So these are all direct answers, easy to answer. Path of greater resistance in transpiration is, so which is going to resist transpiration? Generally, the presence of cuticle prevents the water from evaporating. Only when there is a damage for cuticle, there would be cuticular transpiration. So the greater resistance in transpiration is by cuticle. So the waxy layer which is deposited on leaves and stem, they prevent loss of water. So only when there is a damage to the cuticle, so from that damaged part, transpiration can take place. We call that as cuticular transpiration. If cohesion tension transpiration theory is correct, then a break in water column in a xylem vessel should have no effect at all, increase the rate of photosynthesis, increase the water content of leaves, cause the mesophyll cells to become flaccid, and result in wilting of leaves. So if cohesion tension transpiration is correct, then a break in water column in a xylem vessel should cause the mesophyll cells to become flaccid and result in wilting of leaves. So this is the answer. So if there is a break in water column, so mesophyll cells obviously they don't have water, they become flaccid and the leaves are going to wilt. Okay. In terrestrial habitats, temperature and rainfall conditions are influenced by water transformation, transpiration, temperature and rainfalls are influenced by transpiration. So 14th one, B is the answer. So just imagine, I always remember that whenever I was traveling through this IISc campus, Indian Institute of Science, I don't know whether how many of you have been there, or the Bangalore University campus where there is a lot of vegetation. So even in a hot summer, I never used to feel the heat of it because the temperature would be at least 1 or 2 degrees centigrade lesser. And also I would feel much more cooler there than in the places where there was no vegetation. So this convectional rainfall is also benefited by this transpiration. So that's the reason wherever forests are there, so the rains would be more. So yeah, because of transpiration in terrestrial habitats, temperature and rainfall conditions are influenced by transpiration. So 14th B is the answer. Which of the following criteria does not pertain to facilitated transport? Requirement of special membrane proteins, so which is not pertain to facilitated transport. High selectivity, transport saturation, uphill transport. So in this following criteria, which of them does not pertain to facilitated transport? Facilitated transport is with the concentration gradient, so there would be no uphill transport. So 15th, the answer is D. Uphill transport only in active transport you can notice, not in facilitated transport. A minimum ratio of transpiration to photosynthesis will be observed on a humid and cloudy day, humid and sunny day with no winds. A minimum ratio of transpiration to photosynthesis. On a humid day, the transpiration will be low and photosynthesis will be high because it's a sunny day. So a minimum ratio of transpiration to photosynthesis will be observed on a humid and sunny day. So photosynthesis will be more on that day. So 16th one, so B is the answer. 17th, ascent of sap. This is nothing but uptake of water or transport of water or long distance transport of water is best explained by mass flow, pulsation theory, root pressure, cohesion, tension, transpirational pull is the right answer. Ascent of sap is nothing but long distance transport of water. 18th one, match the column 1 with column 2. This is a little out of the your syllabus. You can notice that Dixon and Joly, so they put forth this transpirational pull theory. Dixon and Joly, fourth one, A4, stomata. So it brings about transpiration. B3, A4, B3. You just see in which condition it is there. A4, B3 in A and C it is there. C, manometer. So manometer is used for measuring so root pressure. C1. So this is the answer. C is the answer for this. A4, that is Dixon and Joly, they put forth this transpirational pull theory. Stomata is responsible for transpiration. Manometer is an instrument used to measure root pressure. Capillary water is only a water available to plants. Potometer is an instrument used to determine the rate of transpiration. E is 5. So that combination you come across only in C. Okay, the 18C is the correct answer. So the maximum transpiration occurs through, is it stomata, cuticle, lenticel? ನಾನು ನಿಮಗೆ ಡಿಸ್ಕಸ್ ಮಾಡಬೇಕಾದ್ರೆ ಹೇಳಿದ್ದೆ ಸ್ಟೊಮ್ಯಾಟಾ is nearly 85 to 90% of transpiration is by leaves, stomata of leaves. 5% is by cuticle, 5% is by lenticel. So stomata is the maximum transpiration occurs through stomata. 19th one, A is the answer. Select the incorrect match with respect to transpiration. So which is the incorrect match with respect to transpiration? So cellulosic microfibrils are oriented radially to open stomata. Green light induces maximum opening of stomata. ABA promotes closing of stomata. Lowering of temperature decreases the rate of transpiration. So green light induces maximum opening of stomata is a wrong statement. So 20th B is a wrong statement. Okay. 21st, stomata are the special structure of the plant or leaves through the transpiration occurs. Stomata of a plant open due to high influx of hydrogen ions, influx of potassium ions. The opening and closing of stomata bandi, they have put forth through a theory, potassium ion pump theory. The most accepted theory is potassium ion pump theory. ಬೆಳಗ್ಗೆ ಹೊತ್ತು ಏನಾಗುತ್ತೆ ಅಂದ್ರೆ ಎಂಟ್ರಿ ಆಫ್ ಪೊಟ್ಯಾಷಿಯಂ ಅಯಾನ್ಸ್ ಆಗೋದ್ರಿಂದ ಸ್ಟೋಮಾ ಓಪನ್ ಆಗುತ್ತೆ. ರಾತ್ರಿ ಹೊತ್ತು ಪೊಟ್ಯಾಷಿಯಂ ಅಯಾನ್ಸ್ ಎಕ್ಸಿಟ್ ಆಗುತ್ತೆ, ಅದನ್ನ ಎಫ್ಲೆಕ್ಸ್ ಅಂತೀವಿ. ಸೊ ಅವಾಗ ಸ್ಟೊಮ್ಯಾಟಾ ಕ್ಲೋಸ್ ಆಗುತ್ತೆ. ಸೊ ಇನ್ಫ್ಲೆಕ್ಸ್ ಆಫ್ ಪೊಟ್ಯಾಷಿಯಂ ಅಯಾನ್ಸ್ ಟೇಕ್ಸ್ ಪ್ಲೇಸ್ ಡ್ಯೂರಿಂಗ್ ಡೇ and that results in the opening of stomata. So 21st B is the answer. Opening of stomata is not affected by, so which of them? Nitrogen, starch, potassium ions, none of these. So opening of stomata is not affected by nitrogen. So A is one which is not affected by nitrogen. So 22nd, the answer is A. 23rd, cytokinin is essential for opening of stomata. Which of the following is responsible for developmental? ನಾನು ಡಿಸ್ಕಸ್ ಮಾಡ್ತೀನಿ, ಪ್ರಾಬಬ್ಲಿ ನೆಕ್ಸ್ಟ್ ಕ್ಲಾಸಲ್ಲಿ ಡಿಸ್ಕಸ್ ಮಾಡ್ತೀನಿ ಅದನ್ನ. ಸೈಟೋಕೈನಿನ್ ಹಾರ್ಮೋನ್ ಬ್ರಿಂಗ್ಸ್ ಅಬೌಟ್ ಓಪನಿಂಗ್ ಆಫ್ ಸ್ಟೊಮ್ಯಾಟಾ. ದಿ ಅಬ್ಸಿಸಿಕ್ ಆಸಿಡ್ ABA ಅಂತೀವಿ ಅದನ್ನ, ಇಟ್ ಬ್ರಿಂಗ್ಸ್ ಅಬೌಟ್ ದಿ ಕ್ಲೋಸಿಂಗ್ ಆಫ್ ಸ್ಟೊಮ್ಯಾಟಾ. Okay, 23rd D is the answer. 24th, conversion of starch to organic acids is required for stomatal opening, stomatal closing, stomatal formation, stomatal activity. It is stomatal opening. So 24th, the answer is A, stomatal opening. Which one gives the most valid and recent explanation for stomatal movement? So D, potassium influx and efflux. It gives the most detailed and valid explanation. The stomata in angiosperm open and close due to their genetic constitution, the force of their habitat, the change of pressure of gases inside the leaf, change in the turgor pressure of the guard cells is the right answer, because of which the stomata in angiosperms they open and close. Guard cells differ from epidermal cells in having chloroplasts. You know that guard cells are also having chloroplasts. Okay, so 27D is the answer. 28th, in the mechanism of opening of stomata, the important factor is it the shape of the guard cells, chlorophyll content of the cell, hormone contents of the cell, protein contents of the cell? Shape of the guard cells. The important factor is shape of the guard cells. So 28th, A is the answer. 29th, which one of the following element in plants is not remobilized? Calcium. ಯಾಕೆ ಕ್ಯಾಲ್ಸಿಯಂ ಮಾತ್ರ remobilize ಆಗಲ್ಲ older leaves ಇಂದ ಅಂದ್ರೆ they are a part of structural component, like they might be present in the middle lamella as calcium pectate. So ಇದು remobilize ಮಾಡಕ್ಕಾಗಲ್ಲ because they are a part of structural component. Okay, so calcium is the answer. 29th ಅಲ್ಲಿ uptake of mineral ions into the xylem is controlled by, is it uh, epidermal cells, cortex cells, or endodermal cells? Endodermal cells are going to control the quantity and quality of minerals to enter into the xylem. So 30th C is the answer, endodermal cells. 31st, a root concentrates mineral by, the root always absorbs mineral by active process because the mineral ions in soil is in lower concentration when compared to mineral ions in the roots. So active transport takes place wherein against the concentration gradient, the minerals are taken from the soil to the root. So a root concentrates minerals by active transport, which is by expenditure of energy. 32nd, minerals absorbed by root move to the leaf through. So the minerals which are absorbed by root, they move to the leaf through the xylem, phloem, sieve tube, none of the above. Minerals ಮತ್ತೆ water, they all move through xylem. There is only unidirectional movement. Xylem ಮೂಲಕ ಹೋಗುತ್ತೆ ಇದು. So minerals also. 32nd, xylem. 33rd, as water and minerals move towards the vascular cylinder of a root, before that they must enter the cytoplasm of endodermal cells. ಸೊ ಅಪೋಪ್ಲಾಸ್ಟ್ ಪಾತ್ವೆ ಆದರೂ ಕೂಡ ಇಲ್ಲಿ ಬಂದಾಗ ಸಿಂಪ್ಲಾಸ್ಟ್ ಪಾತ್ವೆ ಆಗಬೇಕು, ಎಂಡೋಡರ್ಮಲ್ ಸೆಲ್ಸ್ ಮೂಲಕ ಸಿಂಪ್ಲಾಸ್ಟ್ ಪಾತ್ವೆ ಆಗಬೇಕು. Okay, apoplast is through non-living component of cells. Simplast is through plasmodesmatal connections. Which one of the following is correctly matched? Passive transport of nutrients, ATP, ತಪ್ಪು. Apoplast, plasmodesmata, ತಪ್ಪು. Simplast ಬರಬೇಕು. Potassium, readily immobilization, ಕರೆಕ್ಟ್ ಅದು. ಸೊ ಬ್ಯಾಕನೆ ಆಫ್ ರೈಸ್ ಸೀಡ್ಲಿಂಗ್ಸ್ ಕರೆಕ್ಟ್ಲಿ ಮ್ಯಾಚ್ಡ್ ಇಲ್ಲಿ ಯಾವ್ದಿದೆ ನಾವು ಹೇಳಬೇಕಾಗತ್ತೆ. ಬ್ಯಾಕನೆ ಆಫ್ ರೈಸ್ ಸೀಡ್ಲಿಂಗ್ಸ್ ಸ್ಕೂಗ್, ಸೊ ದಟ್ ಇಸ್ ರಾಂಗ್. ಸೊ ಪೊಟ್ಯಾಷಿಯಂ ರೆಡಿಲಿ immobilized, this is the right answer. So 34th C is the right answer. Recognize the figure and write. C andre palisade. Okay, C palisade. D is guard cell. C palisade, D guard cell. Okay, the first two options are having that. A is xylem. Okay, so B is phloem. So A, this is the right choice. C is palisade, D is guard cell, A is xylem, B is phloem. A is the correct answer. The energy source that mainly drives the upward flow of water is light, sucrose, solar heat. Solar heat, it drives the upward flow of water by evaporation. So by transpiration, water is lost. So by evaporation, so the solar heat is the main energy source that drives the upward flow of water. So the foliar absorption of ion helps in rapid recovery from iron deficiency. If it is sprayed in the form of FeSO4, FeCl, FeEDTA, or NaEDTA. So this is a little outside of the syllabus part of it. So foliar absorption of ion. So 37th, the answer is FeEDTA, ethylene diamine, ethylene diamine tetra acetic acid, EDTA. Maximum photosynthesis takes place by phytoplankton, zooplankton, marshy plants, woody plants. Phytoplankton maximum photosynthesis it takes place. Isotopes popularly known to have been used in the study of photosynthesis are, so was it P15C32 or C11P42? C14 and O18 is the correct answer. The isotopes popularly used in the study of photosynthesis are C14 and O18. Photosynthesis is a catabolic and reductive reaction, catabolic and oxidative, anabolic and reductive reaction, catabolic and exothermic reaction. The answer is photosynthesis is an anabolic and reductive reaction. The sequence of energy change in photosynthesis is from light energy, chemical energy is obtained, alva. So light to chemical. So B is the answer. If photosynthesis proceeds for an hour in atmosphere with radioactive carbon dioxide, labeled carbon will be found mostly in sugar and starch. So the answer is both A and B. Both A and B, that is, even in sugar and starch, you find the radioactive carbon dioxide. Photorespiration is otherwise called as C2 cycle. So 43rd, the answer is A, C2 cycle. Phosphoglycolate, it is C2. Photorespiration is seen in chloroplast, peroxisome, and mitochondria. Chloroplast only, mitochondria only, peroxisome. See, chloroplast, peroxisome, and mitochondria is the right answer. So, photorespiration is seen in chloroplast, peroxisomes, and mitochondria. Which of the following is wrong in relation to photorespiration? ಯಾವುದು ತಪ್ಪು ಸ್ಟೇಟ್ಮೆಂಟ್ ಇಲ್ಲಿ ನೀವು ಕಂಡುಹಿಡಿಬೇಕು ಫೋಟೋ ರೆಸ್ಪಿರೇಷನ್ ಅಲ್ಲಿ. ಇಟ್ ಇಸ್ ಕ್ಯಾರೆಕ್ಟರಿಸ್ಟಿಕ್ ಆಫ್ C4 plants. Photo respiration is characteristic of C3 plants, not C4 plants. First statement is tappu. So first statement is tappu. 45th A is the wrong statement, and that is the answer. How many molecules of glycine are required to release one molecule of carbon dioxide in photorespiration? So they have asked this, how many molecules of glycine? Two molecules of glycine are required to release one molecule of carbon dioxide in photorespiration. The receptor in plants that detects photoperiod is a sensory cell, sensory organelle, protein molecule, or is it gradient of potassium ions? So the protein molecule is a receptor in case of detecting photoperiod. So 47th, the answer is C. See, in photorespiration, which is light induced cyclic oxidation of photosynthetic intermediates with the help of oxygen. The substrate is, what is that substrate? So it is glycolate. So 48th, the answer is glycolate.
Sir, glycolate is a product, no, sir.
ಅವ್ರು ಏನ್ ಕೇಳ್ತಾ ಇದ್ದಾರೆ, in photorespiration, which is light induced cyclic oxidation of photosynthetic intermediates. Intermediate ಆಗಿ ಬರುತ್ತೆ ಇದು ಗ್ಲೈಕೋಲೇಟ್. ಓಕೆ, ಸೊ ಫೋಟೋಸಿಂಥಸಿಸ್ ಅಲ್ಲಿ ಯಾಕಾಗತ್ತೆ ಇದು ಅಂದ್ರೆ ಆಕ್ಸಿಜನ್ ಜಾಸ್ತಿ ಇರುತ್ತೆ, ಕಾರ್ಬನ್ ಡೈಆಕ್ಸೈಡ್ ಕಡಿಮೆ ಇರುತ್ತೆ. ಇಂಥ ಸಮಯದಲ್ಲಿ ಸೊ ದಿ ಗ್ಲೈಕೋಲೇಟ್ ಇಸ್ ಫಾರ್ಮ್. And there is also phosphoglycerate. ಎರಡು molecule form ಆಗುತ್ತೆ, glycolate and phosphoglycolate and phosphoglycerate. Okay, but glycolate is formed with the help of oxygen. Photosynthetic intermediates with the help of oxygen. ಅವ್ರು ಅದನ್ನ ಕೇಳ್ತಾ ಇರೋದು. ಸೊ phosphoglycolate is the answer. ಅರ್ಥ ಆಯ್ತೇನಪ್ಪ ಸಿದ್ಲಿಂಗೇಶ್?
Yes, sir.
Photosynthetic intermediates with the help of oxygen. ಅನ್ಫಾರ್ಚುನೇಟ್ಲಿ ನಿಮ್ಮಲ್ಲಿ ಏನು ಮಾಡಿದ್ದಾರೆ, ಸೈಕಲ್ ತುಂಬ ಚಿಕ್ಕದಾಗಿ ಕೊಟ್ಟಿದ್ದಾರೆ. ಆ ಡಿಗ್ರಿ ಕ್ಲಾಸಸ್ ಅಲ್ಲಿ ಇನ್ನೂ ಡೀಟೇಲ್ಡ್ ಆಗಿರುತ್ತೆ, ಅವಾಗ ಅರ್ಥ ಆಗುತ್ತೆ. ಸೊ ಈಗ ನೀವು ಅದನ್ನು ತುಂಬ ಕನ್ಫ್ಯೂಸ್ ಮಾಡ್ಕೊಳಕ್ ಹೋಗ್ಬಿಡಿ. ಫೋಟೋಸಿಂಥೆಟಿಕ್ ಇಂಟರ್ಮೀಡಿಯೇಟ್ಸ್ ಎರಡಾಗತ್ತೆ ಫೋಟೋ ರೆಸ್ಪಿರೇಷನ್ ಆದಾಗ. ದಟ್ ಇಸ್ ಒನ್ ಇಸ್ ಫಾಸ್ಫೋಗ್ಲೈಕೊಲೇಟ್ ಅಂಡ್ ಫಾಸ್ಫೋಗ್ಲಿಸರೇಟ್. ಫಾಸ್ಫೋಗ್ಲಿಸರೇಟ್ ಇಸ್ ಜಸ್ಟ್ ಲೈಕ್ ದಿ ನಾರ್ಮಲ್ ಫಾಸ್ಫೋಗ್ಲಿಸರಾಲ್ಡಿಹೈಡ್ ಅಥವಾ ಗ್ಲಿಸರಿಕ್ ಆಸಿಡ್. ಓಕೆ, ಸೊ ದಿ ಗ್ಲೈಕೊಲೇಟ್ ಅದು ಬಂದು ಆಗೋದು ವಿತ್ ದಿ ಹೆಲ್ಪ್ ಆಫ್ ಆಕ್ಸಿಜನ್. ಇಟ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಫಾರ್ಮ್ whenever the amount of oxygen is more and carbon dioxide is less. Photo respiration takes place in C3 plants. ಅದನ್ನ ನೆನ್ಪಿಟ್ಕೊಳ್ಳಿ. So the product that is formed, intermediate product that is formed is glycolate. So the answer is glycolate. A wastage processes, ಇದಕ್ಕೆ ನಿಮ್ಗೆ ಗೊತ್ತಿರ್ಬೇಕು ಆನ್ಸರ್. ವಿಚ್ ಇಸ್ ದಿ ವೇಸ್ಟೇಜ್ ಪ್ರೋಸೆಸ್?
Photo
Respiration,
respiration.
photos. Yes, photo respiration is the wastage process. Which of the following is not related to photo respiration? See, ಮೂರರಲ್ಲೂ ಆಗುತ್ತೆ: chloroplast, peroxisome, mitochondria. Lysosomes are suicide bags of the cell. They are not involved in photo respiration. So 50th, A is the right answer. Lysosomes. Photo respiration is favored by, ಈಗ ಇಲ್ಲಿ ಬರುತ್ತೆ ನೋಡಿ, high oxygen and low carbon dioxide, high carbon dioxide and low oxygen, high temperature and low oxygen, high humidity and temperature. The simple thing is high oxygen and low carbon dioxide favors photorespiration. So the 51st, the answer is A. During photorespiration, the oxygen consuming reaction they occur in stroma of chloroplast and mitochondria, stroma of chloroplast and peroxisomes. Where are they taking place? The oxygen consuming reaction, grana of chloroplast and peroxisomes. So what is the answer for this? See, it is taking place, oxygen consuming reaction is taking place in stroma of chloroplast and peroxisomes. So they are taking place in the stroma of chloroplast and peroxisms. So this photorespiration, especially the oxygen consuming reaction, occurs in peroxisomes. Glycolate accumulates in chloroplast when there is high carbon dioxide, bright light, low temperature, low carbon dioxide. When does glycolate accumulate in chloroplast? When there is low carbon dioxide and high oxygen, alva. So the answer is low carbon dioxide. So low carbon dioxide. In CAM is a plant in which the stomata are closed during night and open during daytime. So in CAM plants, organic acids are decarboxylated. Organic acids are decarboxylated at day. So that is, they undergo the C3 cycle during daytime and C4 cycle in the night time. So they are separated by time. So in CAM plants, organic acids are decarboxylated at daytime. So 54th, A is the answer. So 55th, in the above cycle, identify the marking A, B, C with their number of carbons. If one ribulose biphosphate, uh, 5 phosphate, 2 glyceraldehyde, 3 phosphate, and 3 diphosphoglycerate, where figures given in the bracket shows the number of carbon in that compound. So let us understand in ribulose biphosphate. So A, so what is the A that is going to form? Ribulose biphosphate ಆದರೆ ಅದಕ್ಕೆ ಮುಂಚೆ ribulose monophosphate ಇರುತ್ತೆ. So this molecule should also be 3 carbon. So it should be ribulose 5 phosphate. For A it is 1. Okay. B 3 phosphoglycerate ಆಗಿದೆ. It becomes 1,3 diphosphoglycerate. So B ಬಂದು 3 ಆಗಬೇಕು. Okay. Then
Sir,
C,
RUBP is 5 carbon, no, sir?
ಯಾವ್ದಪ್ಪ?
RUBP, first one, sir.
RUBP 5 carbon. RM, RUMP, ribulose ಫೈವ್ ಫಾಸ್ಫೇಟ್ ಅನ್ನೋದು RUMP, ಅದು ಮೊನೋಫಾಸ್ಫೇಟು, ಅದು ಕೂಡ 5 ಕಾರ್ಬನ್. See, it is 1 5 carbon. A is 1 5 carbon. So B is 3 and 3 carbon. A is 1 5 carbon. ಈ ಕಾಂಬಿನೇಷನ್ ಅಲ್ಲಿ ಎಲ್ಲಿ ಬರುತ್ತೆ? A is 1 5 carbon. B is 3 carbon. C ಇಲ್ಲಿ ಬರುತ್ತೆ ನೋಡಿ. C is the triose phosphate ಅಲ್ವಾ? So C is 2, that is glyceraldehyde 3 phosphate, and they are having how many carbons? 3 carbons. So D is the answer, correct answer ಇದು. ಸೊ ಇಲ್ಲಿ ನೀವು ಕನ್ಫ್ಯೂಸ್ ಆಗಕ್ಕೆ ಹೋಗ್ಬಾರ್ದು. ಸಿ ಅನ್ನೋದು ಏನು ಮತ್ತೆ ಎಷ್ಟು ಕಾರ್ಬನ್ ಇದೆ ಎರಡು ಜೊತೆಗೆ ಹಾಕ್ಬೇಕು. ಈ 1 2 3 ಲೇಬಲ್ ಮಾಡಿದ್ದಾರೆ. ರಿಬಿಲೋಸ್ ಎ ಇಸ್ ರಿಬಿಲೋಸ್ ಮೊನೋಫಾಸ್ಫೇಟ್, ದಟ್ ಇಸ್ ರಿಬಿಲೋಸ್ 5 ಫಾಸ್ಫೇಟ್. ಎಷ್ಟು ಕಾರ್ಬನ್ ಇರುತ್ತೆ? 5 ಕಾರ್ಬನ್. ಅದು ಡಿ ನಲ್ಲಿ ಕರೆಕ್ಟ್ ಆಗಿ ಕೊಟ್ಟಿದ್ದಾರೆ. ನಾನು 1 ರಿಬಿಲೋಸ್ 5 ಫಾಸ್ಫೇಟ್ 5 ಕಾರ್ಬನ್ ಇರುತ್ತೆ. ಸೊ ಸಿಮಿಲರ್ಲಿ ಬಿ ಬಂದು ಇಟ್ ಇಸ್ 1 3 ಡೈಫಾಸ್ಫೋಗ್ಲಿಸರೇಟ್ ಮತ್ತೆ 3 ಕಾರ್ಬನ್ ಇರುತ್ತೆ. ಸಿ ಇಸ್ the glyceraldehyde 3 phosphate 3 carbon. So C is going to be 2 and 3 carbon. ಸೊ ಇದು ಡಿ ನಲ್ಲಿ ಕರೆಕ್ಟ್ ಆಗಿದೆ ನೋಡಿ ಚಾಯ್ಸಸ್. ಇಂಥ ಪ್ರಶ್ನೆಗಳಿಗೆ ಸ್ವಲ್ಪ ತಲೆ ಉಪಯೋಗ ಮಾಡಬೇಕು, ಮತ್ತೆ ಕನ್ಫ್ಯೂಸ್ ಆಗಕ್ಕೆ ಹೋಗಬಾರ್ದು. ಏನ್ ಕೇಳಿದರೆ ಏನ್ ಫೈಂಡ್ ಔಟ್ ಮಾಡಬೇಕು, ಆನ್ಸರ್ಸ್ ಅಲ್ಲಿ ಇದೆ. ಸೊ ಡಿ ಇಸ್ ದಿ ರೈಟ್ ಆನ್ಸರ್. ಅರ್ಥ ಆಗಿದೆಯಾ ಇದು, ಅಥವಾ ಮತ್ತೆ ಎಕ್ಸ್ಪ್ಲೈನ್ ಮಾಡಬೇಕಾ? ಸಿ ಕ್ವಶನ್ ಅಲ್ಲೇ 1 ಅಂದ್ರೆ ಏನಂತ ಕೊಟ್ಟಿದ್ದಾರೆ ನಂಬರ್. 1 ಅಂದ್ರೆ ರಿಬಿಲೋಸ್ ಮೊನೋಫಾಸ್ಫೇಟ್. 3 ಅಂದ್ರೆ ಇಟ್ ಇಸ್ ಗೋಯಿಂಗ್ ಟು ಬಿ ದಿ 1 3 ಬೈಫಾಸ್ಫೋ ಫಾಸ್ಫೋಗ್ಲಿಸರೇಟ್. ಓಕೆ. 2 ಅಂದ್ರೆ ಗ್ಲಿಸರಾಲ್ಡಿಹೈಡ್ 3 ಫಾಸ್ಫೇಟ್. ಸೊ ಅಲ್ಲಿ ಬ್ಲಾಂಕ್ ಬಿಟ್ಟಿರೋದು ಎಷ್ಟು ಕಾರ್ಬನ್ ಇದೆ ನಾವು ತಿಳ್ಕೋಬೇಕು. ರಿಬಿಲೋಸ್ 5 ಫಾಸ್ಫೇಟ್ ಇಸ್ 5 ಕಾರ್ಬನ್. ಓಕೆ. ಇಲ್ಲಿ ಬಂದು 1 3 ಬೈಫಾಸ್ಫೋಗ್ಲಿಸರೇಟ್ ಇಸ್ 3 ಕಾರ್ಬನ್. And here you are having this 2 3 carbon. 2 is nothing but you have this glyceraldehyde 3 phosphate. Kelvin cycle proceeds in 3 stages, namely carboxylation, reduction and regeneration. CRR alva, carboxylation, reduction and regeneration. So C is the answer, carboxylation, reduction and regeneration. In C4 pathway, the carbon dioxide fixation in mesophyll cells is carried out by the enzyme. ಯಾವ enzyme carry out ಮಾಡುತ್ತೆ carbon dioxide fixation in mesophyll cells? So C, pep carboxylase is the answer. Bundle sheath cells ಅಲ್ಲಿ rubisco, but mesophyll cells ಅಲ್ಲಿ pep carboxylase because it is C4 pathway. Choose the correct combination of labeling the carbohydrate molecule involved in the Kelvin cycle. So one is RUBP. Third one is, here it is going to be phosphoglyceric acid. So second one would be this should be second one. Second is, first is RUBP. Okay. So second is PGA. Third is triose phosphate. D is the answer. ಟ್ರಯೋಸ್ ಫಾಸ್ಫೇಟ್ ಇಲ್ಲಿ ಬರುತ್ತೆ, ಇಲ್ಲಿ ಫಾಸ್ಫೋಗ್ಲಿಸರಿಕ್ ಆಸಿಡ್ ಬರುತ್ತೆ. First stable product formed in the biosynthetic phase in C3 plants first undergoes. So first stable product formed in the biosynthetic phase in C3 plants, first what does it undergo? So does it undergo isomerization, dehydrogenation, phosphorylation or epimerization?
Phosphorylation, sir.
ಎಸ್, ಯಾಕಂದ್ರೆ ಅಲ್ಲಿ ಪಿ ಜಿ ಎ ಆಗುತ್ತಲ್ವಾ, ಇಟ್ ಬಿಕಮ್ಸ್ 1 3 ಡೈಫಾಸ್ಫೋಗ್ಲಿಸರಿಕ್ ಆಸಿಡ್. ಸೊ ದಿ ಆನ್ಸರ್ ಫಾರ್ 59th, ಲೆಟ್ ಮೀ ಕ್ರಾಸ್ ಚೆಕ್ ದಿಸ್ ಒನ್. ಸಿ, ಫಾಸ್ಫೋರಿಲೇಷನ್. ಯಾಕಂದ್ರೆ 1 3 ಡೈಫಾಸ್ಫೋಗ್ಲಿಸರೇಟ್ ಆಗುತ್ತೆ, 3 ಫಾಸ್ಫೋಗ್ಲಿಸರಿಕ್ ಆಸಿಡ್ ಇಂದ ಒಂದು ಫಾಸ್ಫೇಟ್ ಆಡ್ ಆಗುತ್ತೆ. ಸೊ ಅದಕ್ಕೆ ಇದು ಫಾಸ್ಫೋರಿಲೇಷನ್. ಸೊ ಬಂಡಲ್ ಶೀಟ್ ಸೆಲ್ಸ್ ಆರ್ ರಿಚ್ ಇನ್ pep carboxylase, lac rubisco, are rich in Rubisco is the right answer. So bundle sheet cells are rich in Rubisco enzyme, a process that makes important difference between C3 and C4 plant is. What does it make? Important difference between C3 and C4 plants? Photorespiration. C4 plants do not undergo photorespiration. C3 plants undergo photorespiration. 61st D is the answer. In sugarcane plant, 14 carbon dioxide is fixed in a malic acid, in which the enzyme that fixes carbon dioxide is. So, what should be the enzyme that fixes carbon dioxide? Is fixed in a malic acid, in which the enzyme that fixes carbon dioxide is phosphoenol pepcarboxylase alpha. We are talking about C4 pathway here. Sugarcane plant is a C4 pathway. Alpha 62nd, the answer is D. 63rd, the two enzymes which are used for primary carboxylation in C3 and C4 respectively are. So, what are those? RUBP carboxylase and PEP carboxylase. So, the 63rd, the answer is A. 64th, in case of C4 plants, which enzyme fixes the carbon dioxide released during decarboxylation of malate? So, in case of C4 plants, which enzyme fixes the carbon dioxide released during the decarboxylation of malate? Rubisco does that job. So, 64th, it is A. 65th, which pair is wrong? C3 is not a maize plant. C4 it is. Maize plant is C4. So this itself is wrong. C4 Kranz anatomy, Kelvin cycle, PGA, they are all correct. Hatch and Slack cycle, first table compound is oxaloacetic acid. They are all correct. A is the wrong pair. The first enzyme of Kelvin cycle is cytochrome oxidase, phosphopentokinase, RUBP carboxylase, triose phosphate dehydrogenase. So, which is the first enzyme of Kelvin cycle? RUBP carboxylase, that is Rubisco enzyme. Dark reaction of photosynthesis, it occurs in matrix, grana, stroma, cytoplasm. So, dark reaction, it occurs in stroma. The first carbon dioxide acceptor in C4 plant is phosphoenol pyruvate, ribulose 1,5 diphosphate, oxaloacetic acid, phosphoglyceric acid. It is PEP, phosphoenol pyruvate. 68th is A answer. 69th, one molecule of glucose in Kelvin cycle is formed from 6 carbon dioxide plus 12 ATP, 6 carbon dioxide plus 30 ATP, 6 carbon dioxide plus 18 ATP plus 12 NADPH. They are going to give rise to one molecule of glucose. So this is the right answer C. In C3 plants, the first stable product of photosynthesis during the dark reaction is, what is the first stable product of photosynthesis during the dark reaction? Is it malic acid, oxaloacetic acid? It is, the answer is 3-phosphoglyceric acid. So 70th, the answer is C. CAM pathway 71st is observed in pineapple. Pineapple also belongs to the Crassulaceae family. Okay, so the pineapple is the right answer, 71st. 72nd, C4 plants are more efficient in photosynthesis than C3 plants due to higher leaf area, presence of larger number of chloroplasts, or lower rate of photorespiration. This is the 72nd D is the answer. 73rd, synthesis of food in C4 pathway occurs in chlorophyll of. Does it occur in guard cells, bundle sheath, spongy mesophyll, or palisade cells? Where does the synthesis of food in C4 pathway occurs? In chlorophyll of. So it occurs in the bundle sheath cells. C3 pathway ಎಲ್ಲಾಗತ್ತೆ bundle sheath ಅಲ್ಲಿ ಅಲ್ಲೇ food synthesize ಆಗೋದು. So the answer is 73rd B. 74th, in C4 plants, the bundle sheaths cells have thin walls to facilitate gaseous exchange, have large intercellular spaces, are rich in PEP carboxylase, have a high density of chloroplasts. In C4 plants, the bundle sheath cells have a high density of chloroplasts. So that is the right answer. C4 plants are found among dicots only, monocots only, both dicots and monocots you can find C4 plants. 75th, C is the answer. In biochemical phase, fixation of carbon dioxide occurs by Rubisco. Okay. The photorespiration in C3 plants starts from phosphoglycerate, phosphoglycolate, glycerate, glycine. So photorespiration in C3 plants starts from. So 77th, the answer is B, phosphoglycolate. Presence of bundle sheath is characteristic of C4 plants. You find that in C4 plants. CAM helps the plants in conserving water, secondary growth, disease resistant reproduction. It helps in conserving water. Crassylation, acid metabolic pathway helps the plant in conserving water. 6 carbon dioxide plus 12 H2O plus 12 NADPH plus 18 ATP plus 18 ADP it should be. So ATP, 18 ATP and 18 ADP, C is the answer for this. So if you replace A and B correctly, it should be 18 ATP, A is 18 ATP, B is 18 ADP. In CAM plants, organic acids are decarboxylated at day, night, both A and B, none of this. So the organic acids are decarboxylated at day. 81st, A is the answer, daytime. What will be the number of Kelvin cycles to generate one molecule of exos? So how many cycles will it undergo? So it will be 6 cycles. Okay, one cycle produces Kelvin cycle. So they are going to utilize 3 ATP and 2 NADPH2. For 6 cycles, they will be utilizing 18 ATP and 9 NADPH2. Sorry, it is 12 NADPH2. Okay. So just remember, so 6 cycles they'll be undergoing. Which is the first carbon dioxide acceptor enzyme in C4 plants? So it is PEP carboxylase. 83rd, the answer is D, PEP carboxylase. So they are going to first absorb the first acceptor of carbon dioxide. For assimilation of one carbon dioxide molecule, the energy required in formation of ATP and NADPH2 are. So how much for one carbon dioxide assimilation of one carbon dioxide molecule? It should be 3 ATP and 2 NADPH2. So 3 ATP and 2 NADPH2. This is the right answer. Which technique has helped in investigation of Kelvin cycle? So it is radioactive isotope technique has helped in investigation of Kelvin cycle. So 85th C is the answer. What is the outline sequence by which carbon dioxide may be incorporated into starch by photosynthesis? So what is that statement? So RUBP plus carbon dioxide, they form phosphoglyceric acid. They are then going to form exose phosphate, triose phosphate, and starch. Okay, or is it RUBP plus carbon dioxide plus PGA, triose phosphate, exose phosphate, starch? So D seems to be the right answer. So 86th, D is the answer. So it is in the correct sequence. End product of citric acid cycle or Krebs cycle is oxaloacetic acid. So here end product of citric acid cycle or Krebs cycle is citric acid, lactic acid, pyruvic acid, carbon dioxide plus water. So 87th, the answer is D, carbon dioxide and water. Krebs cycle is respiration. Okay, it should not have come anyway. I've given one question, doesn't matter. The senescence of older leaves can be retarded by removing axillary buds and terminal buds. If you remove both axillary and terminal buds, the leaves don't age up. So you can avoid the aging up of older leaves by removing the axillary and terminal buds. In guard cells, when sugar is converted into starch, the stomatal pore closes completely. Okay. Which of the following shows entire plant senescence? Entire plant ages up that you find in wheat, triticum. So 90th B is the answer, 89th A is the answer. Okay, so that completes our discussion for today's paper. Any doubts or clarifications that you have, dear students? ಏನಾದ್ರು doubts ಇದೆಯಾ? Simple paper.